Art historian Werner Spies writes of artist Max Ernst's fascination for the microscopic in works created in 1919. Decades later, Ernst created a series of very small gouache paintings on cardboard, many around 1 inch or smaller, called microbes. The source material for many of Ernst's collages came from popular science magazines such as La Nature, published in the late 19th century. As it happens, during the late 19th century, the theory of symbiosis was getting a lot of attention. For example, in 1867, quote, lichens were shown to be dual organisms, composites of fungi and algae. What biologists had called animal chlorophyll in coral and sponges were shown to be algae living inside cells. One cannot help but think of Ernst's many chimeric and erotic collages, especially during the period of his greatest involvement with surrealism in the 1920s and early 30s. Ernst also created many paintings using decalcomania that contained patterns resembling lichens. One such chimera was Ernst's alter ego, a man-bird creature he named Lop Lop, that appeared in assorted forms throughout his life in Irv. But this only underscores the multitude of animal and plant-animal hybrids he portrayed in print collages and paintings. As Ernst demonstrates, there is a kind of erotics to cutting and pasting. The seamless joins between components can be seen as the join between a man and a woman engaged in sexual intercourse. Coitus is derived from the Latin word coitio, or coire, meaning a coming together or joining together, and is known under different names for a variety of sexual activities. It usually denotes penile vaginal penetration. It is not much of a stretch to see the erotics of the new CRISPR synthetic biology technology in this context. The CRISPR system is widely present in bacterial microbes and used to record a copy of a DNA fragment of an attacking virus into its own DNA. Note that these clones are laid down in chronological sequence, thus assembling an ordered archive of historic assaults. Bacteria can access that record and create a molecule to cut and destroy the DNA of a viral repeat attack. Consider that the microbe now contains a collage made of copies of alien creatures. Additionally, a more beneficial collage hybrid can be created when, quote, phages slip their genomes quietly into the bacterial chromosome and coexist benignly, getting copied along with the host DNA. CRISPR as a human technology matured around 2012 to take advantage of this DNA specificity to preferentially sever specific locations on chromosomes. Scientists can alter the Cas sequence in order to make specified cuts. With this capacity, they can insert, that is paste, new synthetically created DNA at the cut. Owing to its ease, speed, accuracy, versatility, and affordability, CRISPR allows for a range of possibilities difficult to consider previously, including controlled extinction and de-extinction. Such deliberate species manipulations illustrate the human predilection for sculpting the natural environment, creatively crafting an assemblage according to aesthetics and desire. If, as many cultural theorists have drawn attention to, different technologies are extensions of different parts of the human body, then CRISPR can be seen as an extension of the male sexual organ. We can speak of an erotics of CRISPR. All of these biotechnical reproductive insertions are a kind of cross-species sex. It is sexual, sensual, aesthetic, and about desire. As is often the case with the erotic, some people view genetic modifications as perverse. Owing to CRISPR's ease of use and taking advantage of the chronological ordering of genetic insertions, one lab has created a bacteria containing a short movie sequence taken from a Muybridge set of galloping mare photos, thus literalizing the famous quote from William S. Burroughs, "Image is virus."